we're delighted to welcome you back from lunch, and I am happy to remind everyone just to make sure that you silence any devices in case you had those turned on while out during lunch. And with that, I would like to welcome our moderators for the next session. We will be talking about transformative tools to capture early stages of disease, digital health technologies, also known as DHTs. So if we can welcome Joe Hedrick and Sakshi Sardar. Apologies.
Thank you.
All right, we've got the enviable post-lunch session here. So so at the risk of putting everybody to sleep, I'm going to ask everybody in the audience to close your eyes for just a moment. So I want you, with your eyes closed, I want you to imagine that you're hiking along on a beautiful day, the sun's shining, the birds are singing. You step through some bushes and suddenly into thin air. That sensation of falling hits you in the pit of your stomach. Okay, and you can open your eyes now. So you realize you've fallen off a cliff that you didn't see in time, and there really isn't much you can do but hope it's not too tall. For many of us, that's what falling ill with serious disease feels like. Fortunately, we've gotten pretty good at having an ambulance parked at the bottom of a lot of those cliffs so that when we do fall, we can often, but not always, get treatment. In some cases, where we're lucky, there may be a sign warning us or a fence at the top of cliff. But I think anybody that's read the news realizes people walk past signs and right over fences at the top of that cliff. And that's great when those are there because hopefully we can avoid falling altogether, but it's not often the case, and there are a lot of cliffs. So what if in addition to signs and fences, we could build a safety net below the cliffs that people tend to fall off? Or maybe we could have a device that senses when we've fallen and have it deploy a parachute, or better yet, wings that bring us back up or to the other side. So as we've heard this morning, neurodegenerative diseases like Alzheimer's and Parkinson's, or autoimmune diseases like the one I work on, type 1 diabetes, just to name a few, have extended pre-symptomatic or minimally symptomatic stages that present us an opportunity to intervene early and to build those nets or parachutes or even wings. This is where digital health technologies can help, and as Klaus said this morning, help make the unseen seen. And along with this tantalizing opportunity, of course, there are going to be challenges, and we're going to discuss both of those in this session. I'll turn it over to my colleague, Sakshi.
Hello, everyone. Thank you so much for being here for this session. For this session, we will be focusing mainly on how we can utilize digital health technologies earlier in the diseases. And there will be three focus areas. First, what are the measurement gaps when the science can be very subtle? Second, how can we enhance the clinical trial design using digital health technologies? And lastly, how can we provide additional access to diverse population using these digital health technologies? Today, Ashkan, who is the CEO and founder of Biosensics, will be sharing his experience around developing sensor-based technologies and digital health technologies platforms for clinical trial design and research. Pass it to you, Ashkan.
Thank you. Thanks a lot, Sakshi. It's a great pleasure to be here. Good afternoon, everyone. So let me start with, yes. So as a quick introduction, Biosensics is an FDA-registered medical device company focused on developing wearable sensors and digital health technologies, mainly for clinical trials and research. This includes our wearable devices, for example, for precision actigraphy, falls, if somebody falls the cliff, gait and balance, instrumented activities of daily living, as well as other, for example, motor symptoms monitoring, as well as digital and video-based assessments, for example, for looking at cognition, voice biomarkers, facial characteristics and ptosis, hand and finger movement, and gait and balance. In addition to providing these technologies to pharmaceutical companies, we also conduct our own clinical studies with the objectives of developing digital biomarkers and digital tools for different diseases. Currently, we have 17 different priority therapeutic areas where our efforts are supported by more than $20 million of funding from the National Institutes of Health. They are in collaboration with multiple top research institutions as well as patient advocacy groups. So in the next few slides, I'm going to actually show you a selected set of results on ALS, Parkinson's disease, and progressive supranuclear palsy related to some of the points that was highlighted or going to be highlighted in this study. Starting with our ALS study, this is the case that the participants come to the clinic at baseline and then every 3 months for the total of 12 months. So there are 5 different visits. During each visit, we collect all clinical data such as ALSFRS, and then they go home wearing a pendant and 2 wrist sensors in order to measure their physical activity, posture, falls, as well as upper limb function. They also receive a digital tablet, which they use in order to conduct a speech, handwriting, pattern tracing, and fine motor control assessments every 2 weeks. So this is a previous design of our tablet that is shown here. Our new generation looks much nicer. So just to show you some results related to how these technologies can be used in order to assess clinical measures and clinical outcomes, starting with speech measurements and looking at voice biomarkers in order to assess 2 different critical subdomains of ALSFRS-R. One of them is the bulbar and the other one is respiratory. As you can see on top, you can see multiple speech features such as articulatory rate. They are highly correlated with the bulbar subscore of ALSFRS-R, which is typically used as the primary outcome in ALS clinical trials. Also, for example, in the case of a specific task, in this case phonation ah, where patients actually essentially keep saying ah as long as they can. Again, you see good correlation between what is measured by the speech biomarkers versus the clinical score, which is the ALSFRS bulbar subdomain, respiratory distress subdomain in this case. Now moving on to sensor-derived measures, again in ALS, what I'm showing here is the analyzed data from our wrist sensors, which essentially it quantifies and measures upper limb function, hand function, how much I use my hand during activities of daily living and with what quality. So what are the qualities of movement of my hand as, for example, I grab a cup of coffee. So again, you can see very good correlation with what is being measured, in this case for 7 days, that's on the y-axis, versus the clinical score on the x-axis, which is the one of the upper extremity subdomain of ALSFRS. And then looking at the patterns of physical activity, such as daily walking duration, as well as number of sit to stand transition. Again, you see very good correlation. This is perhaps the best one, as you can see in the screen, in terms of correlations with the clinical scores. Now, in addition to this, we developed predictive models where essentially the clinical score is predicted by the device itself. For the sake of time, I actually don't have them here. Moving to the next study, this is a study in progressive supranuclear palsy and Parkinson's disease in collaboration with Mass General Hospital and Johns Hopkins. In this case, this is again another multimodal data measurement using our DHT, digital health technology products, where the patients wear sensors at home. They also receive a tablet in order to, for example, provide their fall diary, do psychomotor tapping tests, conduct, for example, a speech and cognition test, and also do their gait assessment at home. So this is an example of an individual with PSP, progressive supranuclear palsy, conducting a gait assessment called Time Up and Go at the home of the participant. So obviously, the deployment of this device at home enables collection of the data at a very high frequency there. This was conducted in this study every month. So if you look at this slide, what you see on the left-hand side on top is the variation in the clinical score as a function of time. So essentially, that is PSPRS, PSP rating scale, as measured in the clinic by a clinician as a function of time over 12 months. The lower plot is essentially what is measured by the sensors at home at a much higher frequency every month. And what is shown on the right-hand side is essentially the change in these clinical outcomes, clinical and sensor-derived outcome. So the clinical outcomes are gray, which you see a smaller slope, a much more significant variance compared to what is measured by the sensor, which is blue. So what this means, for example, is if you design your clinical trials based on the outcomes that are derived by the sensors, you would need a smaller cohort in order to look at the efficacy of the drug. So instead of having 100 PSP patients in each arm, you might need 70 or 75, as an example. We also looked at speech measures. So again, looking at the speech measures in both Parkinson's disease as well as PSP. You can see multiple speech measures can differentiate Parkinson from PSP, as shown on the top plot on the left-hand side. Speech measures also show significant correlations with the clinical scores, as shown on the bottom one. And then on the right-hand side, we developed machine learning-based models. These are predictive models in the sense that they can essentially, just from a simple speech task, can provide the dysarthria score for the patients in PSP. As the next step, what we have done is we actually expanded our speech analysis in multiple neurological disorders. Identification of the disease is one of the key challenges in neurology. So in many cases, for example, individuals with PSP is diagnosed as PD. Same story with multiple system atrophy and other Parkinsonian disorders. So what is shown here on the x-axis is, on the y-axis is the true label. So these are confirmed patients with PSP, PD, ALS, Huntington disease, and control patients. And what is shown on the x-axis is what is predicted based on our machine learning models, where you can see, for example, our ALS prediction has a recall of 92%, very accurate. Also, PSP and PD in general are predicted better than a general rural clinic without specific expertise, for example, in progressive supranuclear palsy. I want to highlight that also, because we are talking about early stages, we have papers that show how these speech measures as well as sensor-derived measures can differentiate, for example, prodromal Huntington from controls, as well as assisting in early diagnosis of dementia. So I'll stop here and happy to answer any questions after the panel. Thank you.
Okay, great. Thanks very much. I think hopefully people saw a couple of things in that presentation, hailing back to some of the conversations that we had earlier this morning. I think also in there, one of the things that we do see is that particularly in early disease, a lot of the symptoms in clinical science can be subtle or even not very visible. Biomarkers can be really important in these scenarios to try and understand what's going on. However, they need to be, as we discussed earlier, anchored in clinically meaningful metrics. And that might require long-term follow-ups in certain scenarios. So if we think about how we use DHTs in the earlier pre-symptomatic diseases in this context, one of the questions that we have is how should developers think about how to anchor what their DHT is telling them to clinically meaningful metrics, and then what key technology aspects of digital health technologies need to be considered to provide the supporting evidence, quality supporting evidence, to support their use as drug development tools. And I think what we'd like to do is have the panel really be conversational, but maybe we'll kick off the conversation with Michelle.
I'll take the first half, you take the second half of that question.
Sounds good.
So for those of you who have never seen Annie and I together, we are a tag team.
Including our shirts.
Including our shirts, which is the tell me your context of use, which is an important recurring theme. A recurring theme of this discussion today in DHTs. And so I think it's a really great question because when it comes to digitally derived endpoint opportunities, we are seeing the emergence of how can digital health technologies be used as we go into earlier disease, right? Or before, hopefully really we're showing that symptomatic aspect of where a clinical diagnosis would be given at that time. And I think with that, we have to really plan accordingly, because we may actually not have that natural history of what early looks like. And I'm going to use PD as an example. Where's Diane? Can I? May I? Okay. So the Parkinson's Consortium has actually been looking at this for the last 4 years plus of actually trying to collect longitudinal data as we shift earlier into PD, right? And so all of the guys out there who are in the PD space, I think you know what I'm talking about, right? We're seeing this shift in options of trying to go earlier. And so efforts have been ongoing is, well, what does that look like, right? And how do I actually know that's early, which I don't think we actually have a definition of that, right? Which that's another topic and another day. But what has actually been important with that in a couple ways is, one, qualitatively talking to patients about their lived experience. Now, in the PD and probably in a lot of diseases, as we go early, the small subtle signs were not noticed, or a realization of maybe that is my beginning of my lived experience now with this disease diagnosis. And so what has happened in that effort is that we've gone to folks who are just more recently diagnosed, who can reflect back on what that was like up until they got to diagnosis, to kind of hone in on, oh, that subtle change in my phonation, in my voice, was actually probably suggestive of something. So first, it's really talking to patients and getting a lived experience. What do they remember about prior to that diagnosis, up to what they can, to kind of help us understand what that was. Because if we're going to shift early, we're going to have to capture things that are early, right? And in PD, it's probably not going to be some of those hallmark motor symptoms, perhaps. The other piece is that what we did, and I'm not saying this, we may not need to do this forever, but at the time when we were doing this, is that in the PD space, we also had to understand what was actual normal human aging and experiences, and so because there may be overlapping. And I think if you hear people talk about their diagnosis, they're like, well, it's a wait and see, right? Or it's a process of elimination, right? It wasn't this, it wasn't that. And so we had to understand that. So in pediatrics, we talk about understanding typical childhood development, and how does that underline? Well, we kind of have to do the same thing when we get in the adult side, is what is actual natural progression of aging, and how does that tie in? And then the other piece is actually what's been extremely beneficial is just the longitudinal aspect. And so I think collecting longitudinal data, kind of understanding this, is going to be really helpful as we go into these earlier diseases, to help us then identify what are going to be the things we want to capture in our trials. What can we actually capture, and be able to translate that as a digitally derived endpoint. And so we really do want to leverage then our PFDD guidance series on what we need to do as an endpoint. Because if it's going to be supporting efficacy, we still have to follow those good principles. But I'm going to turn to Annie more about on the technology piece and what's needed for that, because that's also the flip side, right? There's two pieces to it. So I'm going to ask you about the second side.
Yeah, and I think that's also where you really should be looking at the PFDD guidances in concert with the DHT guidance for remote data acquisition and clinical investigations, because the two really do work together, because you need to come up with whatever's the meaningful aspect of health by talking to patients, do your measurement science of like, what's the concept, tell me your concept of interest, and then using the technology, can the technology, the DHT, measure what you think you're measuring, and then you need to understand that it can do that accurately and reliably. And with the potential for novel measurements too, I think one thing, you brought up anchoring. There may not be an anchor to, like, back to PD, when you're talking about later stages of PD and UPDRS versus an earlier stage, you might be talking about completely different concepts. So you don't need to necessarily try to anchor to the gold standard that may not really be the gold standard. But that's a whole different topic. But really thinking about is the technology measuring what you think it's measuring, and then is it measuring that concept that's going to get you the information you need to either be a digitally derived endpoint. In some cases, they actually may be diagnostic devices that actually come into CDRH, and so you also need to think about all of those aspects as well. And so that's really where you have to really bring together all these different groups. So that's, I think, the piece where you see with like CPATH and other groups about collaboration so key, because you need the people with the experience with the disease, you need the measurement scientists, and you need the tech developers, because we don't want, and we see this, I joke, I'm an engineer, so I can make fun of engineers, but you don't want to just develop tech for the sake of tech. So, having a real clear intentional purpose of using digital health technologies, and why are you using the digital health technology? Is it actually benefiting the patient? Is it benefiting understanding the disease or condition? Is it benefiting leading to an earlier diagnosis or treatment?
Yeah, I think that's why like Annie and I being together is because we have the engineer and the measurement person saying you've got to work together.
Yes.
We have to work together. We work together. And so we all need to, as we're exploring these technologies and their utility.
Yeah, I think those are some great points. Ashkan or Scotty, I don't know if there's anything you want to add or a question that you may want to raise in regard to this.
Oh, Joe, I've got questions. I think it's worth, PD has obviously been central to a lot of what we discussed today, and I think it's worth looking at the Watch PD effort, which Diane was obviously pivotal in making happen. And some of the learnings there that might map towards the question, really, in terms of what can we identify in that kind of prodromal, early, pre-symptomatic stage. And I think of the many things that I think were learned, and this is as an observer, not been involved and not an expert on this. I apologize to Diane this morning before I made the inevitable mistakes I'm about to make. But one of the things that really leapt out to me was that in the symptom of bradykinesia is that it seemed that the algorithm linked to the multiple sensors that were used in that study was able, over the 12-month period of assessment, to do a better job of notifying the increase in severity of that bradykinesia beyond what the clinician was able to do. And that's the whole concept behind Watch PD, is to say, there are challenges with the gold standard measure. You would agree. But there's other opportunities, other ways. And that signal was identified. And I think there was an observation in terms of arm swing change as well, which is another thing. The thing about bradykinesia is that in reading some of the literature related to that, it seems to be something that can start up to 10 years prior to actual diagnosis of PD. So if we think about the question there, is that if we've got at-risk groups, at-risk individuals, what can we be doing with a digital health technology to start picking up on things at the very earliest stages? So work that CPATH has been involved in is already starting to look at that particular angle, I think.
So maybe calling back to some of those pilot type studies that we were talking about in an earlier session here. Sakshi?
So building upon that, all the comments around, how do we build the drug development tool side of things, right? Something that you mentioned is some of the signs or symptoms can start way earlier, right? And it also ties in with the stages of the diseases, right? Earlier stages of the diseases. So as we are trying to go earlier and we are trying to develop these tools, what do you think about characterizing the population and the subpopulation so that we understand how these signs and symptoms are manifested, so that now those characteristics can be used for developing the necessary tools?
So maybe I can add something here, also add maybe in response to the previous question as well. So when we look at these early phases, and I have to say I'm an engineer, I have a PhD in mechanical engineering, so I'm not a clinician, but have had the opportunity to work with, I guess, many great clinicians in different disease areas, specifically in neurology. When we look at a disease like Parkinson, which is not rare, I think obviously developing these tools, I don't want to say are easier, but essentially there are more number of patients. There is much more that is known about those, that, for example, about PD, and there is much more data available. So, for example, for us, one of the challenges we have is when we get to these early stages for rare diseases. For example, for Huntington disease, then all of a sudden the pool of the number of patients that you have access to becomes much smaller. The studies become much more expensive. We are not sure if we get to the N numbers that FDA would require. So that's one of the challenges I see, that when we get to rare diseases, essentially being able to collect the data in order to back up your hypothesis. For example, whether the speech goes first in Huntington disease. And In some of those diseases that are progressing slower, again, it becomes more challenging. Again, I use Huntington because we have some direct experience with. Typically, Huntington is a slow progressing. So again, that's another challenge that gets created because you need to monitor patient for a longer period of time, whether they're early stage or not, in order to get any signals. In general, when we look at these, so I guess in response to your question, it's really, again, in my view, and based on our experience, is disease specific. Most neurological disorders have some overlapping symptoms, but again, there are symptoms that are somewhat unique or different to different neurological disorders. So in order to pick up the right DHT, essentially, we need this collaboration between engineers and mainly clinicians, patient advocacy groups, in order to start the conversation and coming up with the right set of hypotheses that then we need to collect the data in order to verify and validate.
I think as we want to go early, I think this is why we have to work in a pretty competitive space. I don't think we can accomplish what needs to be done solely in the time frame of a drug development program, right? Because we still may need some of that longitudinal information to do that. So if we can start in the opportunity as a pre-competitive space, I think that is going to be very beneficial to hope the overall aspect of the work. I also think what you have heard earlier today is that this isn't in isolation, right? We should be also advancing the science and other things in biomarker development, in the translational science. Is there an opportunity with enough information to develop a staging system, for example, that can help us identify patients maybe earlier? These measures can help tie that in and fill it in. These are typically iterative processes, so the idea is that it's going to be a living document and grow over time. So I think that's important, is that we shouldn't be solely just focusing on DHTs. I think we have to be, as we talked about in COAs, we need to use a lot of things. I think in these earlier aspects of disease of understanding, we're going to have to use multiple drug development tool approaches to help fill in as we're learning. I think in the rare disease space, I do think there is an area of opportunity with digital health technologies to measure concepts that we just don't measure well any other way, right? In ALS, we struggle with the Balber stuff, to be honest. We really do. And I personally think that DHT is going to probably help us with some of that, get us a better measure than that COA, than that clinician-reported outcome, right? Because that's what I hear from patients talk about, right? I can see it visually when I see that, but we need to be able to quantify that in a way. And so I definitely think that because we are limited in sample size in a rare disease space, though there are opportunities where a DHT is going to definitely be helpful in the things we just really can't capture well. And so really making targeting and understanding it. What do we know about the disease, or if it's part of a family of diseases? Like you said, some of these neurological examples you gave all have similar things, and we hear about how it's kind of like a process of elimination to make sure which one I have. Well, I think that's where, could that help us with that, some of that differentiating? Could this be informative, as Dr. Brockie said earlier with her question about how this research and regulatory and clinical practice all tie in together? Could this help us, to Annie's point, the diagnostic piece? Earlier diagnosis from some of these things. So I think we have some great opportunities. I think we still need to be patient on what are they going to, which is the right fit, right? I think everyone sees this as the answer to a lot of solutions, but I think we still need to be thoughtful. I think we do have good examples of digital health technologies that are being used to support other things right now. And I think it's still a great opportunity for us, but we have to make sure we have some patience to make sure we get the sufficient data to be able to make those conclusions.
I think I'll just add that, it's beyond just the pre-competitive consortium. We also need to think about our models for data sharing, information sharing, so that we can all learn from each other. So if there are similarities across different neurological diseases, we don't need each group to be doing their own thing. Like, let's learn from each other, and then also the access to data, especially when you're talking about AI, like you need data and you need enough data to, both test and train your models. And so we really need to think more broadly about how do we think about data sharing overall for longitudinal information to actually be able to model and understand and leverage DHTs.
Can I add amen? This is at the heart of so much of what we do. I don't want to lose too much track from the discussion there, but it astonishes me, to be honest, that there's not more recognition, acknowledgement of the need to do this as a collective. It's at the heart of what CPATH is, really. I've always kind of seen CPATH as the tide manager in terms of trying to lift all the boats at the same time. But there are multiple examples of where I think there's been good work done on a consortium level. IdeaFast, which you might be aware of, the IMI-funded initiative, which was looking at, I think, fatigue, activity, and sleep patterns. I think it's a European Commission-funded effort, which is, I think, wraps up in a couple of years' time, but it's done a really good job of trying to acknowledge the patient requirement of that, but also trying to look forward to developing the measures that can be used on a collective for consensus basis. And we have activity going on within the CPATH, within the PIRO consortium was looking at that within chronic heart failure, for example. That actual project has a activity monitor-based endpoint as part of this effort, going through the COA qualification program. I'm sure if anyone wants some background on how intense that is as an experience, my colleague Sonia Eremenko will be more than delighted to debrief you all. But I've been with the organisation for 3.5 years, and it's amazing to me that the scientific rigour, the integrity of the work being done cannot be questioned, but it is a very slow-moving process. We've got knowledge of that. So in recognition of things like the resource availability at something like the agency, it doesn't look like, I asked you a year ago if you could clone yourself. I'm guessing that
I
hasn't
haven't figured that out yet. I don't think we have an approved mechanism for that either.
I don't think anything's coming to CBER yet for that.
We haven't cloned myself yet, unfortunately, or Annie, or some of my other colleagues that will be here today in the next couple days, who will talk about all of these different opportunities in drug development. Unfortunately, we haven't cloned some of ourselves yet.
We can wait. But,
we can work on that.
I'm assuming you haven't cloned yourself,
No.
of course, because how would we know? But the point being is that I think there needs to be that recognition is that there is a finite resource. It is, and we all want a piece of our FDA expertise, but it's a stakeholder, it's a discussion, and what you told me some time ago, Michelle, was that you're here to help come to solutions. It's not just about being the wise, iconic head at the table, passing judgment on everything that's going on, which maybe you envision yourself, I don't know. But I think we need to recognise that if we come together, if it's within obviously a common context of use, and we know what the specific meaningful aspects of health, then if we are doing these things as a collective, then we are going to move forward more comprehensively.
Yeah, I'll just take a little bit of the liberty to comment. I think that's exactly right, Scotty, and I'll just want to acknowledge that there are a lot of companies that have worked with the T1D Consortium at Critical Path that have given the data from their failed studies. In fact, I think every major study that's been done in the new onset space, certainly, and most of the ones in the prevention space, companies have been willing to come and say, yeah, we even if they're not part of the consortium, yeah, we take the data, help try and understand what's going on and really advance the science. So I think the more examples of that we see, maybe the more encouragement we can give to these companies that, look, at the end of the day, you're only helping yourselves and, more importantly, helping the patients.
Well,
really.
I'll just add to that, Joe, about why it's important to data share, or please send us your data and your IND. If you don't want to data share, I encourage data sharing. It's because that's how we learn too, right? That's how we're going to learn from the data. That's how it was going to feel comfortable with the data that you submit. What is it really doing? What is it really measuring? We can bring our internal expertise if it comes in the IND and the NDA space and look and see, right? I get that there's risk. I get that you may feel that's not ready for prime time. But that does give us some opportunity to be able to provide better advice going forward, not only to yourself, but to the field as a whole, where we're looking at this. And I also think in the pre-competitive space, I know that there's probably some anxiousness about technology and things like that, or the data is maybe not pretty, right? It's too noisy, it can't get out. That's how we learn. And that's where in the pre-competitive space, we can collectively learn that way. How do we handle noisy data, or how can we provide advice on better data collection opportunities, on some of that. So I think that's why it is important. I understand the hesitation. I think, I think folks are comfortable with maybe the trial data in the more traditional sense of trial data, right? But this is kind of new, and we still have some unanswered questions collectively in the field, but we're only going to learn if we can do it together. And I really would encourage, again, I get the comfort level thing, but sharing somehow that information so then we can feel, because I hear a lot of people going, well, FDA won't accept this, right? We hear, Annie and I hear this all the time. You won't take this, right? It may be the first time I've heard about X, right? That you're yelling at me that I'm not accepting, right? Okay, literally, something that happens all the time, and I'm like, well, that sounds fascinating. But in my head, I'm going, I've not even heard about that in X disease or in X program, right? Because it was never said, right? It might have just been happening type thing. So please do talk to us about this, because we are interested as well with how these data and these technologies can help advance drug development.
And I will say too, from the device perspective, as we've been reviewing, DHTs that are medical devices and AI enabled medical devices, the more we're seeing, the more comfortable our review teams are, the more they understand it, the more less training we have to actually do. And it also lets us evolve and change or see what we need to do from a policy and guidance perspective based on, what we're seeing and learning. And maybe something we thought we needed earlier, we realized we don't need that. Like most of the time, we're probably not going to need the raw data from the DHT because one, we have no one really knows what to do that. You need some processing and you need the algorithms to figure out what to do. Otherwise, you've got 20,000 pages of code and data that no one's going to be able to interpret. So, those are the types of lessons, and those are the things that we're learning as we go over time. And so that is where we really do encourage people to come talk to us early and often. That's our refrain, certainly.
So maybe I want to add something about this notion of conducting this in a pre-competitive environment. I think one of the parts that might be missing is the technology provider, and as an example is us, right? So if you think about data sharing, right, the raw data that is being shared, yes, that can be pre-competitive, but as soon as you analyze it, it becomes bound or dependent on our IP in a sense that, if you run the same speech with someone else's algorithm, most probably you get a different result. So that's, I think, one of the challenges with regards to making this pre-competitive. The other challenge that we have faced as a company, we are a small business, we have less than 30 people, is to bringing people together to fund a pre-competitive study. So, in most cases, we essentially moved forward ourselves. That's the reason we have 17 priority therapeutic areas. We work with Allison, for example, on CMT, at least to collect the data, get to the point that typically we can get whether funding from one pharma to expand it or whether get NIH funding. So those are the two challenges I wanted to highlight from the pre-competitive space. One of them is you can't go from one solution to another because they're not going to be 100% compatible. And the other one is this pre-competitive space is still, I don't see a lot of interest from pharma to come together to, for example, in some of the rare diseases that at least we have experience with, to fund a larger study. So I just want to highlight those points.
So, if I may add, I think it also stems from the fact that these are extremely resource-intensive processes. Yes. For pharma, they are trying to develop a drug, and then trying to develop a digital health technology is like building a road to get to a place, right? So, from that perspective, the pooling of resources can be helpful. But then I also understand that not all the disease areas would have the same kind of opportunities. There are some disease areas which are very relevant for a lot of people, so they are very easy to bring together a lot of stakeholders that are available for public-private partnerships. But then when we go for certain disease areas, we don't have a lot of interest from different stakeholders, then it becomes a little bit challenging to pull together those public-private partnership kind of constructs. So building upon the discussions that we have been having, earlier stages of the disease, trying to set the context in the earlier stages, right? And trying to identify what will be the application in that context of use, and characterizing the disease such a way that we can leverage that characteristic for developing that solution. I wanted to hear more about your views on how we can use these digital health technologies in optimizing the clinical trial design, right? We have touched a little bit upon the metrics that might be relevant, but then how do you use it for maybe sample size reduction or for selecting patients? Any thoughts on that specific aspect of using digital health technologies? And I agree with Michelle that DHT is not a solution for everything. It has to be appropriate for that specific solution.
I think we've had conversations within the ECOA Consortium membership around how baselining could be supported by DHTs. But I think there's something of a presumption in that, and I don't want to go too much down a rabbit hole, but the type of technologies available and suitable to do this is something that we kind of wrestle with in the ECOA space. We've had a lot of discussions about the concept of bring your own device, right? Where you can use a standard smartphone to download an app to complete a patient-reported outcome online. The uptake of that has been curiously low, even though it seems to make a lot of sense from a patient centricity perspective. So without going too high level on the discussion, when I think about where you can use something like a sensor-based wearable that you would have to provide to a broad set of potential patients that you were looking to characterize the disease more finely, to assign them to a particular arm, it does get particularly complex, I think. And my kind of dream, in many ways, is that we had broadly available commercial devices that hit the standard in terms of being appropriate to measure a particular set of metrics, a particular, measurements, whatever they may be, that would allow us to make those decisions. But that's, I kind of feel like Star Trek, Dr. Bones, that thing you used to scan people with. I thought, that's what we want. We need one of them. And Michelle would love to qualify that, wouldn't you?
I think standards is very important, and
Yeah.
we are starting to see that with different groups, and we are, at least FDA is part of a lot of different standards development organizations, and for example, with the OTC hearing aid final rule, we actually do point to a CTA standard on, hearing aid performance. So there is opportunity through some of those types of venues to actually try to do some of that, so we can actually get to, maybe having a certain threshold or baseline, and if you conform to that, then maybe we have some confidence in using the DHT for, whatever specific purpose. And I don't want to interrupt the rest of your thought.
No, I was going to sort of build on that concept a bit to refer back to the chronic heart failure work that I referenced earlier, where that's where we're going to come to soon. Because you're thinking about an activity monitor, there's a lot out there. I've got this one, it's random and generic, it's not Samsung and Apple. I'm trying to be radical with my choice of wearable. But we have to develop those standards so we can get as broad a set of individuals involved in, and chronic heart failure is a massive issue globally, right? Anything cardiac is. And we've got real opportunities. We've got genetic predictors that we can look at a certain set of people and think, if you've got a device that is to a certain standard and a certain quality level, we should be able to tell an awful lot of things about where you belong and where you could be potentially best treated at early stages as well, to come back to the whole concept of this session.
Yeah, and I think also the opportunity for decentralizing aspects of a clinical trial and bringing it to where people are, instead of people having, especially for people in populations who don't have access to an academic medical center readily, the ability to actually be able to collect data at home, maybe even should lead to hopefully more diversity in the clinical trial. And so I think there's a lot of opportunity there. There's still a lot of work to get to that point. I know there's a later session on AI, so I'm not going to touch too much on, but there is work happening, and I know companies are using AI for like site selection, optimization, some of those aspects of clinical trials.
Yeah, I was going to bring up decentralized trials, which is one thing that I think a lot of people are hoping that DHTs can leverage. And as a reminder, the DHT is software and a hardware, right? So there are things with Scotty and the ECOA Consortium and the vendors to do that, and that's been around for a really long time, and we know how successfully to implement that. Well, can we uptake some of the leverage there and some opportunities, since it is a DHT, to help decentralize trials even more? and understanding of what is needed to do that, and again, standardization and things like that if we're trying to move clinic visits to home, or what can I capture more at home that I don't need that big long clinic visit. We hear this a lot in the rare disease population. I would say anyone with a chronic condition, right? To go in for a 2-day, 8 hours each day, I've tried to travel 600 miles to get there, right? I'm exhausted, right? We hear this, we hear it from families that I'm not going to perform that well when you've given me 10 performance outcome measures to do, right? Now, if you've come to me and I've given you comments, I'm going to tell you not to do 10 performance measures if you've come early and often for advice. Or let's think about this strategically of what's my critical things I need to capture in those visits. But I think decentralized trials, we see this as an opportunity, but we still have to do the good homework. We still have to standardize. We have to understand what we're capturing. Can we capture this at home? What does it mean? So you still need to do the homework for it, but we definitely think that that is an area of opportunity.
And rare disease in particular. I mean, we have a project which we're doing in collaboration with Lindsay Murray, who was in the session prior to lunch with the Rare Disease COA Consortium, around video-based COA in rare disease specifically, really driving at the huge burden that taking a patient with a rare disease to a hospital or a clinical site involves, and trying to negate that and use technologies, DHTs, to try and get the information we need, but doing the necessary homework, as Michelle says.
Yeah, it doesn't even have to be a rare disease. I mean, even if you're physically close to the location, 2 hours in traffic, if it's your child, that you're a caregiver, you're taking your child out of school, for a half day or a whole day. I mean, yeah, these are huge burdens that I think DHTs
People work.
Yeah,
Yeah,
They take
well,
their
and
time off work.
Yeah, absolutely.
Yeah.
So, yeah, I think this is one of the promises that we really need to strive to understand, and then how to contextualize that in the clinical trial setting.
So maybe I can give you 2 quick examples. So one of them is the study that I showed on progressive supranuclear palsy. It was a completely decentralized clinical trials for the parts that are, there are clinical visits as needed, but what the decentralized part enabled essentially a much higher frequency of data collection at home, which a much less burden. And in that specific case, essentially a clinician was calling through the tablet in order to supervise the data and even not only assess, for example, gait using our device as speech cognition, but also assess the clinical measurements over telehealth. So it's a much higher frequency of data collection with much less burden, especially with someone like, with a disease like PSP. The other point I want to mention is an example of one of the clinical trials that we have done with one of the large pharma companies, where they deployed our technology in the clinic, but also at home. And one of the thought was the patients most probably will not come to all of the clinical visits. So what the DCT part enabled is, if they were not coming to the clinic, they were showing a pop-up on the tablet that at least they would collect the data. So in this case, the risk of missing data was much lower, because even they were supposed to come to the clinic, at least at the parts that they were not coming, they were collecting data. And this was a clinical trial. Yeah.
Sakshi, I'm going to make you one of the panelists for a moment. I know you've thought a lot about some of these most recent questions here in terms of trial decentralization and conduct of trials. I don't know if you'd like to add a few of your thoughts?
Sure. So when I was thinking about this session, we are trying to go earlier in the diseases, and the decentralization part is where we are trying to do passive monitoring, and in certain scenario, continuous monitoring, which can be feasible if we are using, or if you are repurposing some of your already existing devices, like your watch or a wireless sensor that you might have in home, and then it is continuously collecting the relevant metric for a specific disease area. But then when you think about certain other disease areas, we might not be able to have that kind of a deployment, right? And in those scenarios, how would the decentralization passive assessments would look like? I don't have the answer for it, but it is something that we should also think about, because we don't want to do passive continuous monitoring 24/7 10 years earlier into the diseases. So I think there is a gap there that we need to think about how we can solve that and identify what can be appropriate levels of assessments, right? Does this happen every year, 6 months, for how many days, so on and so forth. And again, depending upon the technology as well, right? With digital technologies, you can do a lot of things, but then a technology might not be relevant for certain signs or symptoms. So I think there are a lot of developments that need to happen if we want to go much more earlier in the diseases. We are trying to develop things where the signs and symptoms are being manifested, and we are still trying to solve the use of digital health technologies rather than the unmet need of the disease area. And if we have to go even further, 5-10 years, what would that entail? And I think we need to understand that that is a very complex task as well, and we should start thinking about it now so that when we get the necessary information, the characterization of disease, we can leverage it to create the necessary drug development tools and then use those for optimizing trials as well.
So I just want to interject one thing, and I know we're talking about early disease in here, but I saw Pat Furlong earlier today, and I have a memory from going to a meeting with PPMD back in March with the boys, and they're a little bit older, and they were saying, "Don't forget about me." And so I know we're talking about early and going early in disease, but we should remember if we are in the slowly progressive, neurogenic, autoimmune type landscape, and while yes, we know that we will have better long-term outcome results if we go earlier, there still may be an unmet need population that these technologies could helpful and could help them maybe spur potential innovation and development in some of those areas as well. So I just want to throw that in there, is that while early is important, and I think it's a very important conversation, because we know that long-term those outcomes are better in the long run for things. We still may have some areas in groups where there's some unmet needs that we may be able to get and focus on, that these technologies and these drug development tool opportunities, we shouldn't forget about that as well, because they could be just as informative to help us with that disease understanding to continue to go early.
Yeah, I think that's a great point, Michelle. I mean, these diseases are continuum, and we can learn from one stage and apply it to an earlier stage, or the converse can also be true. And I expect we'll see some of that particularly in some of the neurodegenerative spaces as well. And how do we help patients that are already somewhere along that trajectory today, right? I don't want to say to somebody, it's too bad we didn't catch you 10 years ago. Maybe we could have gotten you in, and we've seen that in some of these trials where, well, we'd like to get you in, but honestly, it's too late. So one of the things I want to get to here, and then we want to make sure we have some time for the audience. Terminology in this space is really a challenge. People use different things to mean the same thing, or they use the same word to mean different things. Scotty, I know this is particularly a bit of a bugbear for you. And so I wonder if you'd like to address that and maybe mention some of the resources that are out there for people to make sure that they're using terminology in a concise and standardized manner.
Yeah, and thank you for allowing this question to be on the table, because it's something that particular interest to me. We're at a stage right now that in the United States, I hope I'm right in this conclusion, but I read it in a paper that was just published last month. We still haven't had a drug approved that used a sensor-based DHT to support primary endpoint. Okay? And we've got to think about, that will come, it will happen. There's reasons for that, but one of the reasons, I think, has been certainly discussed extensively within the ECOA consortium and the PIRO consortium as well, the sponsors and the ECOA consortium as the vendors, is that lack of alignment, particularly globally, but also even cross-divisional, if you will, or cross-functional within sponsor companies. I'll give you a great example, and a very illustrative one, is that the Mobilized initiative, which many people will know about. Again, an IMI, EU-funded initiative, which has kind of finished its first phase, developed what they call mobility outcomes, but they classify them in the European context as biomarkers. If you look at them through the US FDA lens, it's a performance outcome measure. It's a COA. So here we have a large, substantial, successful collaboration that, in being driven from Europe, looks at something that is a fundamentally different qualification pathway than what it would be here. That's not right. Jurisdiction should not be dictating the terminology that we use in terms of ultimately developing tools that allow us to get medicines out there, better medicines, faster. And there are initiatives out there. We've got to acknowledge the work, again, the FPA, the European Pharmaceutical Alliance. I don't know the exact acronym, I'm afraid, but they made a very substantial call to arms about trying to align on digital endpoint related or digital biomarker related terminology. While ECOA is kind of like on the borders of that, we're in collaboration with the PRO Consortium. We've developed something called the ECOA Lexicon. Version 4 is in development right now, where we're trying to get everyone involved in ECOA space to use the same language. We're all talking at least fairly common things. But there's still, yet last week, I noticed that something called the Digital Health Technology Assessment Consortium, have you heard of that? I did as of last week, have just issued an appeal to individuals to help them develop a lexicon for DHTs. And that's the kind of thing you say, yay, and what? At the same time. So we have to come together. And I think, in all sincerity, in the context of DHTs, there's a PPP opportunity here for us to start bringing stakeholders together. Terminology is one of those things we can build on. And CPAF is the type of organization that can do that.
Well, I think with that, I want to give the audience a chance to, if they want to sneak in a couple of questions here before we thank our panelists. If you just raise your hand so the folks with the mics can find you. And this gentleman here.
I'll just shout and you'll hit the question.
Oh, Mike's coming right behind you here. Mike with the mic.
To what extent do you have ongoing dialogue with NIST? Because you think about the commercial sector, right? You get to standardize the technology, like it's been saying, so watches, wristbands, whatever you use in the ring or a ring. Is that a conversation that you can influence within AST?
I'm going to go with you,
Yeah,
That's for you.
we talked to NIST quite a bit. And then, like I said, we are FDA's members of, we're members of AMI, ISO, CTA, all of the big, large standards organizations. So it's an ongoing effort. We actually have a very large standards program, especially in CDRH.
Yep.
Thank you.
I think this will be our last question, unfortunately. We're getting
Oh, wow.
our time flag, so go ahead.
I hope it's actually a significant question then. I just want to bring back one of the previous question that I don't think has been fully answered. So there's no doubt that the role of digital health technology can help reducing sample size, increasing power, and all of that. But how about early stages of diseases, right? So that actually was a question, and I don't think we really got a full clear understanding of how can we use digital technology when we have not yet developed a biomarker, right? So we're looking for a biomarker using digital technology where clinical cannot really help yet, right? So, and we heard many times this morning as well, be focused, which is, precision, and be reliable, which is basically the basics of any biomarker, right? So how can we use digital technology where we haven't yet developed a biomarker and in the early stages of disease? And you mentioned Parkinson's, low-hanging fruit, yes, in later stages, but in early stages, we don't have yet anything. So anything there, any pearl of wisdom?
I think in the landscape we're at right now, we're going to have to probably start with where is the current clinical diagnosis and then trying to work our way backwards, unfortunately. I think that's where we're at. I think you're highlighting why we are emphasizing the need, so we can't do the DHTs and the biomarker development, the COA development, all that in silos and separate. They're going to have to probably be actively occurring at the same time simultaneously, and hopefully those groups are helping with each other. The understanding. I think the other piece that's really critical that we really haven't mentioned a lot about in this session, though it is very much relevant, is what is my understanding of the disease? Do we have a natural history? Do we understand the pathophysiology? Those are all important things too that will help us. So I think we have examples. Again, we use the PD example as one. I'm hopeful those lessons learned from that effort will help identify the things that you've highlighted as the current, I don't want to call them barriers. I think they're the challenges that we have to face right now. I think we're going to overcome those challenges from the early adopters and innovators who are trying to explore this in some of our diseases, so that we can then inform our smaller diseases that don't have the population to do that, but we'll have those good lessons learned of some of those approaches. So I don't have probably the best advice for you. We very much acknowledge that as a challenge, but again, I think we can overcome it from the lessons learned from those innovators and early adopters of these technologies to help us better inform. So, because that is reality.
Well, I would also add, I think you can also learn from diagnostic development. So we just saw this with COVID, over the counter. Everyone can swab up your nose and all of that stuff. That's a new concept. That's a new way of doing it. So, you know, let's learn from how it worked, novel development, and the diagnostic space is a clear area where there's a lot of lessons learned about how to measure something new in a completely different way.
Ashkan or Scotty, anything to add to the answer there?
Amen.
I'm sensing a theme, Scotty.
So, you know, for example, we have done some studies that, as I mentioned, for example, in Huntington disease, we showed that at least some of the characteristics and features as collected by sensors and speech are very different in prodromal compared to controls. So there are some studies, many studies done actually by other groups, including in prodromal Parkinson. I just want to highlight also one of the applications that might be somewhat relevant of these DHT is as a screening tool. So, you know, typically you have exclusion-inclusion criteria. You would say, you know, are you within this age range, within, for example, male, female, etc. You can collect very simple, for example, a speech, or, for example, do passive testing that are relevant to fine motor control or cognition, to at least use it as an aid to supplement that kind of screening. So I just want to add that that's also relevant to early phases, in my view.
And I think, to acknowledge the question, I still think we're very early in the journey with DHTs. We've got a lot to learn still, but I think broadening our vision to think about the early stages of disease progression is something that we should be thinking about now, which I think was the point we were trying to make. But we need to do a lot more in terms of getting DHTs out there, sharing the failures, sharing the data, being honest about when it works and when it doesn't, harmonizing on regulatory pathways. We've got a long way to go. We're making good progress.
I think that's a great point, Scotty. I mean, I mentioned to Sakshi earlier today, there's a saying I love: the best time to have planted a tree was 10 years ago. The second best time is today. And so thinking about how to bring some of these DHTs into your early trials to begin to gather some of that early data, even if it's not part of your outcomes for that particular study, and again, bring these discussions to the regulators, share them with critical path, let us help to try and understand some of these things. I think maybe we can make progress a little bit further and not always be wishing we'd started this 10 years ago. So I think with that, we'll thank our panel for a great discussion here today, and we'll turn it over to the next session.